Hello everyone here on site and also online. It's very nice to be here. Thank you for the invitation and thanks Jere, and yeah, very nice to be here and have a chance to present some of our work. Very modest but still some studies that we have been doing dealing with heat, cold and hypoxia. And initially I thought that I would be spending most of my time discussing hypoxic things, but it turned out that I'll say a few words in the end about hypoxia, but I was filling the presentation, so it will be mostly heat and then cold. We have quite a lot of unpublished data that I want to show to you. So then I have only 30 minutes, so I needed to cut the hypoxia part. Anyway, and now it would be mostly resting state studies, but not solely, but mostly. But so indeed, we've been doing a lot of exercise physiological things. So I'll start with those just as an introduction. So what has been probably, I would say, our main focus is to study the acute effects of exercise and long-term training effects of exercise training in different tissues in human body. So we've been mostly focusing on heart and skeletal muscle, I would say, but also studied lungs, bone and bone marrow, kidneys, liver, adipose tissue also quite a lot, to some extent skin as well, and nowadays more and more brain studies. We have many going on. So indeed, exercise has been the thing. And here is our typical study setting. This is a bit fast now. It's because it's been quite invasive. Very many, and as you can see, quite a lot of devices. So the scanner, so it's a positron emission tomography scanner. It's not good for anatomy, but really good for functional studies, physiological studies. And then we combine. So breathing hypoxia, other devices measuring here. Would be nice to do a good whole body exercise in the scanner, but that has not been possible. We have done a supine cycling exercise studying heart. That's the closest we've been getting about real exercise. So what we've been doing is this one leg kicking exercise confined to quadriceps femoris muscle group, but still it's exercise and it's quite nice. It's confined to these muscle groups, and then hamstrings are inactive. You can figure out many things by that. One thing also in the past has been that we've been utilizing quite a lot of cannulations. So typically we've had a venous catheter here in this arm, and then the radial artery cannulation, and then many times also here cannulated femoral artery and vein, and taking directly blood samples from those, and also infusing drugs like adenosine, phentolamine, nitric oxide inhibition things. So really challenging the circulation and metabolism, and then studying what happens. So and there's been like a blood pump what we need for these investigations, because this is the tracer we've been using a lot, the radio water. So 15 oxygen labeled radio water, and we need the arterial input function, which is described here for the modeling, and then we get the tissue time activity curves. And the more these curves are aligning with each other, the higher is the blood flow. That's like the basics. But that's now it's better with new scanners like whole body PET scanner, because then you have heart there. You can get the input function from there, from heart directly. But still invasive though. So but this is a little bit fast. And we're being muscle specific in many of those studies. So some years ago, so Bengt Saltin, Ylva Hellsten contacted us. Hey, we want to do a study with you. So here's Bengt Saltin taking femoral venous blood sample at the same time that when I'm taking arterial blood samples. This was the nitric oxide inhibition study where we could show for the first time in humans that when you inhibit the formation of nitric oxide, muscle resting oxygen consumption increases. So it directly inhibits mitochondrial oxidase C in the mitochondrial and lowers oxygen consumption. When you take that away, then the oxygen consumption increases. One of the good things to be very muscle specific. And some of these studies, exercise studies, I was presenting This is just a follow-up to the heat studies we've done. And the first one was, I was giving a presentation the American College of Sports Medicine meeting in San Francisco 2009. And then Craig Randall was the chairman there in the session. He's the thermal scientist globally, I would say. And then he came to me after the session and said that, Ilkka, I want to do a study with you. And I said, okay, yeah, let's do it. But we don't have any money. So he said, but don't worry, we have. So that's how it eventually was happening. And then because they had, like, I don't remember whether they had published that study, most likely not yet, but they had some indirect evidence from Copenhagen with Michael Sander, Bente Stalneck, so that this is xenon clearance study, so that when you locally heat the leg, in this case calf, there was an indication that also muscle blood flow might be increasing due to heat stress, because for decades before that, the idea was, based on research, that it's only skin, like the textbook knowledge, it's only skin, no increase in skeletal muscle blood flow. But so these studies started to challenge that assumption. So this is what Craig wanted to do here in Turku, and this is what we did, because he knew that we can be very muscle specific, only in the muscle. So this was the study. So here are positron emission tomography images from the cross-sectional slide from the calf. Craig wanted to specifically study calf because their findings are also from there. So let's not go to thigh, for instance, just do the same location. So the more red and yellow you see, the higher is the blood flow. And then, yeah, obviously more dark and blue, lower is the blood flow. So so Craig brought this, very many other devices as well, all the temperature probes and all that. But one was this heating suit. So subjects were basically wearing this suit that we then heated. But we first heated only this calf, only left leg calf. And what you can see here, compared to the normothermic baseline condition, is that blood flow obviously increases quite a lot in the skin, but also in the muscle. If you see, it's basically nothing compared to exercise, real exercise, but the thing is increases. So that was the point. So we could directly show that also muscle blood flow increases when you are heating persons. But then Craig had a really good idea, like later I figured it out. So he wanted to do then whole body heating, but then we left the other leg covered, not covered that. Just to, okay, we systemically heat, but not locally heat this one. And the thing was, so the skeletal muscle blood flow remained higher, elevated here, which was still also locally heated during whole body heating. But then there was no increase in this muscle, in this calf, which was not heated. So not just like indirect systemic heating was not leading there to the effects. So then obviously if you go to sauna, so then I don't know, maybe you are heating it locally. I don't know what happens, but this is what happened in this study. So also bone blood flow was increased during the heat stress. So that was also quite interesting. I'll show you. We've been continuing this bone blood flow measurements also later. And when we did this study, I was later I've been regretting that, because usually we take also a lot of blood samples, but here we didn't really take. So later we've been doing a lot of immunological studies lately. There's a flagship program, really big one in Turku, led by Sirpa Jalkanen Academia, concentrating all the immunological aspects in different diseases. And we've been studying immunological effects in cancer patients in response to acute exercise. But now also lately we just published a sex difference study about immune cells between males and females in response to vigorous exercise. So that's something. So I was keen that, for instance, these heating responses, I would like to know what happens with immune cells. But now luckily we have had a study going on with Jari Laukkanen. So results from many years ago, but now eventually we have a manuscript in revision. Not guaranteed that we are going to publish it based on the comments, but we'll see and try. But anyway, so here we studied, so there is, like for the total leukocytes, but also other like lymphocytes, neutrophils, other eosinophils, basophils, granulocytes, some increases in response to sauna heat stress. We also did a cytokine measurements, so we see some good significant mean responses with the immune cells, but not so much with cytokines. But interesting thing is that then with the cytokines, we see very many correlations with the change in body temperature and the change in cytokines, which is to us interesting, even if we don't see like a robust changes there, but still. Okay, so this is basically everything that I wanted to say about heat stress things. Some old ones, one new one that we've been doing. Then going towards cold stress. So in our center, this is now been a big interest for several years, pretty much since we did this heat stress study with Craig, because Kirsi Virtanen, so 2009, they published this really nice study in New England Journal of Medicine showing that humans, adults also have brown fat, which is like activated in cold and like in contrast to white fat, which stores energy. So brown fat is burning energy. So I think this must be the least amount of subjects. A study published in New England Journal of Medicine, they had only 5 patients. But 3 of them, they took also biopsies. So from here, this location, they could really show that this is brown fat, all the UCP expressions and all that. And also in response to cold stress, glucose uptake in brown fat was increased very much. Actually, yesterday when I was preparing that, I noticed I did not previously pay attention, but also in white adipose tissue, there is an increase in glucose uptake. So it's not only confined to brown fat, but obviously it's the highest there. But so Kirsi has a lot of things going on in response to cold exposures, but not only for the brown adipose tissue, but other tissues as well. So they were, in a way, they were the first ones to really show this, but actually in Oulu, quite a few years ago, so in early 80s even, so they've been studying these workers that are outside, working outside. So here's a 48-year-old timberman taking biopsies. They also saw brown fat there. Same thing with this 25-year-old lumberjack, brown fat documented also there. So there has been these indications also previously, but the other study was the really most robust one. So they also got to the good journal. But it's not only about brown fat. So Dennis, how do we say the surname? Blondin? Oh, Dennis Blondin. We say it like this. Yeah. That's an active research group also in Canada, also working with Kirsi in collaboration. So they also study brown fat, but they also study very holistically, so also muscles. So, and what is coming from their studies is that it's not like either or. So it's the combination. It's for the cold-induced thermogenesis. It's the brown fat, but also skeletal muscles, and then the combination that the heat production happens. So that's like the background. And then Warren Gregson. So this is a group from Liverpool. And Tim Cable, Warren Gregson, so when they were Qatar football, soccer, world championships some years ago, so already at least 6 or even 8 years ago, so this group from Liverpool was recruited to Qatar to build up all the physiology department and training centers and all that there. So then Warren called me some years ago that, Ilkka, hey, I want to do a study with you. So then I say that, yeah, let's do it, but we don't have money. So he also said that, don't worry, we have. So that's the study I'm going to tell next that we did with Warren Gregson and Tim Cable and Chris Morvini. So in this study, it was more basic physiology, but obviously they say it's very hot there, so it was for the recovery purposes as well. So we had 3 groups, 10 males in all of these groups, and then we did also a little bit more later. So we did different degrees of cold exposure, cold water immersion. So not complete, so they were covered here. So basically mostly legs and few other parts also there. 8 degrees, 15 degrees, and 22 degrees. And then we had a resting study first, then a little break, and then a very heavy exercise study. And then right after, they went to this cold water immersion situation. There we had the proper control condition without any different temperature water of the exercise, but Warren wanted that we don't do a proper control for the resting part. We want to know what happens 8, 15, 22. And this also with Craig, we measured muscle temperatures with the needle. We also did this with Chris with 1 centimeter, 2 and 3. And then when we were doing this, I was also saying, why don't we go just bone deep? Would be good to know what happens there. You'll see why we've continued that. So yeah, temperatures, then they swallowed also this pill. We measured internal temperature, skin temperatures, and then also skin blood flow, different groups. And we tried to pay a lot of attention so that so you can see the wheelchair. This is pretty much the most important thing, so that they don't activate the legs and muscles at all. So we minimized that. So we lifted them from the water also to the hospital bed table, and then with the wheelchairs, we're carrying them to the scanner and also help them so that there's no activation. So that, because we did, I can show you, I don't know if any one of you recognize the person there, but he promised that that's okay to share. And so he was one of the subjects. So this is the condition. So this wasn't next to the scanner, so we needed to move them a little bit, but we wanted so that the legs are staying cool during that period. So that was pretty much the setup. Okay, so then the results and also imaging. So this is now blood flow image and then MRI image from the middle thigh region. So now we are here in the thigh region. So here the region of interest. So we here also focus on the quadriceps femoris muscle group. And I'm sure you know and remember the anatomy, but for the result section, I'll just point out you so that this is the vastus intermedius muscle that is deep here, just above the bone, which is rich in capillaries and type 1 muscle fibers, also like postural muscles. So for the results, I'll just mention this is vastus intermedius, because what happened that in response to these cold water immersion protocols was that if we had looked only this global quadriceps femoris muscle group as such as a bulk, we did not detect really much there, no changes. But then look what happens with this vastus intermedius muscle, so deep muscle close to the bone. In response to the, when you get to the colder water, 8 degrees, there is I would say, huge, but at least to some extent, quite an increase in muscle blood flow. Increase, so not decrease. People would obviously many times, and what we see in the more superficial muscles, maybe not so much more consistent in this vastus medialis muscle here, but in the other muscle groups, blood flow in the superficial parts decreases, but increases there in the deep muscle. So obviously different responses. And what we think, what might be happening here is that I'm not sure, because it's really hard to measure, like shivering, you can measure with EMG here in the superficial muscles, but not so much be there in the deep muscles. So we don't know whether some sort of shivering also started to happen there, and then the metabolism was increased, and then the blood flow was increased to meet the demands. And this is not only based on our results. So Dennis, these results, what they published in response to whole body cold stress, is also that they see much higher glucose uptake in the deep muscles compared to the more superficial muscles in the body. So also supporting the idea that they're in the internal parts, body probably tries to preserve the heating. Also same results based on Kirsi's group, what they have been measuring, also oxygen consumption in brown adipose tissue, white adipose tissue, and in these different muscles. And what is their major conclusion also in this study, that it is so brown adipose tissue is a minor and deep muscles are a major contributor to cold-induced thermogenesis. So, and it seems like that our results are in line with that if you combined all of those. So maybe good to try to go as deep as possible. So I won't show you, not to confuse you too much, not the individual muscle responses about post-exercise results. So, but here also the global quadriceps femoris muscle group responses. I think the title here already says it all. So what is the main result here is that after exercise, cool water immersion reduces post-exercise quadriceps femoris muscle perfusion more than cold water immersion. So also here, it appears to be temperature dependent. And I don't know, so the physiological and clinical meanings of these, I don't know, and also for the recovery purposes. But then it's for if you want to cool your muscles and reduce muscle blood flow, if that is the aim, so maybe then it doesn't need to be so cold water. And temperatures appear to make a difference. So now we've tried to go deep, and then this is probably the deepest we can go. So now we have a follow-up study. We've analyzed here the same study setting. We've analyzed also bone and bone marrow blood flow. What is happening there when you are cooling these muscles? So we are quite confident that we can be only in the bone and then bone marrow, measure those. And then you would probably, and I think also our research group agrees, it's the idea that yeah, definitely we would see a vasoconstriction and then reduction in blood flow. But it appears to be a little bit similar that was in the deep muscle. So maybe not so strong responses. So with the 22 degrees compared to the 15, maybe some trend towards reductions here in both bone and bone marrow. But here, 8 degrees compared to 22 degrees at rest. Remember, we didn't have the proper control condition. So there, blood flow appears to be increasing in bone and bone marrow. So this is what we find. And then I don't know, but again, for the rehabilitation purposes, if you have an injury, I cannot say that what to really suggest. Would it be the cold water or more temperate water? But this is something that probably needs to be figured out later. And then I think also, so this is post-exercise results. Also still yet unpublished, but also the 8 degrees compared to 22 degrees increase in blood flow, and then here 22, and here we have the proper control. There bone and bone blood flow, there is a reduction in bone blood flow. Most likely, so this is probably not just the direct cooling effects here, because this especially 8 degrees, it triggers a response, so sympathetic response. So that's probably so there's a lot of things activating and probably preserving the temperatures and activating. We are not quite yet sure physiologically what is really happening, but that is my idea, that this sympathetic response is somehow modifying these responses. Good, so then we were smart enough eventually to take some blood samples also, so arterial blood samples. And here, so we've also done quite comprehensive metabolomics with mass spectrometry, and also cytokine panel, close to 40 cytokines we measured. And I won't go into the details. This is still work that we are working on, but looks like, so there's not so much happening with the metabolites in response to these cold water immersions. but especially 8 degrees right after 10 minutes after this exposure. So there's already quite a increase in many of the cytokines. And then later also this 15 degrees water temperature kicks in, and then there are also alterations in cytokines. So we see much more responses. So these cytokines are these, let's say, mediators of immune cells that are actually mediating many of the effects of immune cells. And then the direction is that most of the cytokines, we see a reduction in those, not an increase, which is also interesting. Yes, so then I have a few more minutes, I think, if we save some time for questions. So I have only, actually 2 slides, but maybe one that, so I tried to collect, so here are some of the hypoxia studies we've been doing. We have not been doing training studies, long-term training studies, but acute hypoxic studies. Also they combined exercise, but then at the resting state. At some point, I think I came to Juha also when we were initiating these studies, so Juha has been also on board. So how to build up this system with all the things that we could eventually go there. So we've been addressing skeletal muscle blood flow and metabolism, then also measuring blood flow in adipose tissue and also in bone, and then studied heart, what is happening in all of these tissues, also some preclinical work in regards to heart. And so my summary based on our studies, and I think they are quite well in line also other studies that have been performed in Copenhagen and USA and other parts. So I would summarize it so that if the hypoxia is, let's say, not too extreme, like 300,000 meters, so at rest, even if arterial saturation is a little bit lowered, we don't yet see increases or changes in muscle blood flow. The thing only that is happening is that when the arterial saturation and oxygen content are reduced, there is a similar reduction in the venous venous oxygen saturation. So and the oxygen extraction, it's not increased yet there. It's just similar. It's reduced to the same extent as is the reduction in arterial oxygen saturation and content. But then with exercise, submaximal intensities, there is an increase in blood flow. Also maybe a little bit increase in oxygen extraction. So, but then muscles during submaximal exercise, maybe this is even more clear when you have only localized muscle exercise, when there's no competition with the other muscles, maybe a little bit different condition. But muscle is quite fine in terms of oxygenation. So the blood flow increases to the same extent, so they compared to the normoxic condition. So the oxygen supply is quite normal, pretty normal to the tissue. But then obviously when you increase the exercise intensity, you start to reach your limits in cardiac output, maximal one, and then everyone has a capacity, a certain capacity in that. So, and then it's not even quite sure if you can reach the same cardiac output in hypoxia than in normoxia. And so you eventually, because you have less oxygen content in the arterial blood, eventually you reach your limits earlier. And what happens also when we've been measuring, it was over 5,000 meters, so 11% of oxygen we gave to the subjects and measured the cardiac function, also coronary blood flow. So there is so what body wants to do also during exercise, preserve so that heart and brain are well oxygenated. It doesn't really care so much about the other organs. So that would probably be my main summaries based on the hypoxia studies we've been doing. But then I'll show you. So this is, I think, beauty of that we have had these arterial and venous cannulations over the leg when it's been doing exercise. So here's authentic data. So we've been doing these in random order. So obviously here are arterial values in normoxia. As you can see here, oxygen saturation is 99. Oxygen content 208 milliliters per liter, and here are the venous values. So this was performed later than the hypoxia condition. So you can see during exercise, hypoxia arterial oxygen saturation in these subjects being 85. So quite some reduction. But then cannot even what the saturation here. Here's the saturation 27. But the thing here is, so when athletes also, maybe that's the point I want to make here, because we measure here also pH, so the acidity. So also in this case, so the subject venous, what is coming from the leg is 4.4. So someone would say, okay, it's already quite some increase from rest. I didn't have here now the normoxic condition, so cannot directly compare. But if you look at the venous acidity values, it's exactly the same. 7.30 and also here 7.30. So just to also say, even if there is a reduction in oxygen saturation, athletes are so worried about, oh yeah, it's pH is changing so much, and they measure lactate and they say, oh, acidity was so much, it was so high. Based on, I mean, we don't really see. So that's why. So would be, yeah, if you talk about acidity, maybe it's good to measure it rather than try to figure out something just based on the lactate values. This is maybe the most important point. I just wanted to show authentic data that it's really been cool to have these values also directly from the exercising leg. What is the situation? Not just some general value taken from somewhere else. Good. Thank you very much for the attention. I'm happy to answer any questions you might have or try to answer.